My name is Rachel Kaplan, and I'm from Fairbanks, Alaska, which is very far from the ocean. But I imagine, like many of us here today, I've always been fascinated by the animals that live in the ocean and how human systems can both harm and conserve ocean ecosystems. And therefore, I'm now a PhD student at Oregon State University, where I'm part of both the Geospatial Ecology of Marine Megafauna Lab, where I am today, and the Zooplankton Ecology Lab. And that means that I get to spend a lot of time thinking about both whales and their prey. And I'm really excited about this work, as I imagine many of you are, because you're here at 10 a.m. on a Saturday morning, which is wonderful. And so I'd actually like to start off by hearing a little bit about you. I know that you're a group with a lot of background and expertise in all sorts of cetacean research and matters. And I would love if a person or two wanted to unmute and share kind of some of your main interests and what you're thinking about with whales right now. And if others just want to put something in the chat, then I'd love to see that later. Is there anyone who'd like to share?
Well, I can tell you a little bit about the American Cetacean Society. We're the oldest whale conservation organization in the world, started in 1967. And the primary focus of ACS is the conservation of whales, dolphins, porpoises, and their habitats. And we do this through public education, through funding research grants and conservation actions. And we have citizen science activities. We have our speaker series like today with lectures. We have journals and newsletters to help educate people, and looking at some of the problems that whales are facing in the world today.
Thank you so much for telling me about that. I think that the work you do is really interesting and important, and I'd love to hear about others' personal interests as well. If you feel like putting something in the chat as the presentation goes on, then okay. I'll be really happy to look at that later.
This is Cheryl, and I'd like to add to Joy's comment.
Great.
Joy, Roger, and myself first became active in learning about whales by volunteering with the state of Oregon's Whale Watch program. I know I did it for 16 years. They did it similar. And at one point, we kind of dropped out of that and formed the Oregon chapter for the American Cetacean Society. So from there, we have expanded to our knowledge instead of just being informing tourists that came to our state, but helping them to spot whales and trying to educate them to a certain extent. We've been educating ourselves now through the speaker series, etc.
That's so cool, and I'm so glad that I get to be part of your speaker series, so thank you for inviting me. I want to focus today on an issue that I imagine we're all pretty familiar with, which is whale entanglement. And these events are a problem in much of the world, including Oregon, as we know, but we can use scientific approaches like ecosystem modeling to try to mitigate this problem. And so in the project that I want to talk about today, we're trying to bring in a new element to these ecosystem models, which is prey data, in order to best predict whale distributions. And I think that some of you may have attended Dr. Solange Derville's recent talk about entanglement and this work. And I wanted to share some of the ways today that we're building on that work. So just a little look at where we're going today. I want to talk about the prey that's so important for whales, the ecology of the Oregon coast a bit, whale entanglement, and some of the policy surrounding it, ecosystem modeling as a process and a tool, data collection, how we get the inputs that feed these models, both about whales and their prey. We'll look at some acoustic data together and talk about future directions of this work. And I'm really excited to share with you today, not just about my research, but about the experience of being a graduate student and doing this work, because it's a huge privilege. So I thought we could start by all taking a moment to imagine that we're standing someplace on the coast of Oregon that we love. This is a picture from the God's Thumb hike in the Lincoln City area, which I did recently and totally stunned me with its beauty. And we actually even saw a whale blow from our hike. So we all know that the abundant marine wildlife is one of the things that makes the Oregon coast so special. And we'll talk about some of the reasons why this area is so rich today. And I especially want to focus on one of the things that is most important to many types of wildlife here, which is krill. Krill are tiny ocean animals less than an inch long that live in dense swarms that contain thousands of individuals, just like the one we see here. There are 2 species of krill off the coast of Oregon, and there's some important differences between these 2 species, which we'll go into a bit more later. Both types are an essential prey item for so many animals off the Oregon coast. And these next few slides will give us a look at the oceanographic underpinnings of why that is. So you can see on this map, here we are in Oregon, that one of the dominant oceanographic features that controls our coastal ecosystem here is the California current. So this is one of the world's eastern boundary currents. And these are really important systems because even though they comprise less than 1% of our total global ocean area, they account for about 5% of global marine primary production and about 17% of global fish catch. And one of the reasons behind this incredible productivity is the process of coastal upwelling. So this is how it works. In the spring, we get northerly winds off the coast of Oregon here, and these initiate what's called the spring transition, which is a really crucial transition point for this ecosystem. So as these winds come down the coast, the rotation of the earth causes displacement of water offshore, and this causes the upwelling of cold, deep waters. And these waters from depth contain high concentrations of nutrients, which bring this pulse of needed elements into the ecosystem and results in a lot of productivity reverberating up the food chain. So one of the first results of this upwelling pulse are blooms of phytoplankton, like the diatoms we see here. These are these beautiful single-celled algae with silica skeletons, and the animals that graze on them, like copepods here, also tend to really thrive after a big diatom bloom following upwelling, as do our krill. And this productivity at these lower trophic levels reverberates through the food web and is really important to the marine megafauna species that I think many of us here love. So that includes seabirds like marbled murrelets and auklets, as well as seals and of course whales. And there are also a number of human systems that interact with these marine systems. So there are several commercially important species like rockfish, lingcod, and salmon that are the basis of commercial fisheries. This is a picture of the working waterfront in Newport, and all of this infrastructure is really a testament to the productivity of this coastal ecosystem and how the Oregon coast is built around that. And there are also a lot of wonderful opportunities for recreation and tourism, like you've mentioned already today. So as we see around the world, the intersection of human and natural systems can also present challenges. And one of those that interest myself and my collaborators, and I suspect all of us here, is whale entanglement. So I want to spend just a minute talking about that issue, both globally and here in Oregon, and some of the policy that attempts to mitigate this problem. So entanglement, such as in fishing gear, has been identified by the International Whaling Commission as the biggest human-caused threat to cetaceans around the globe. And here in the United States, entanglement is considered a take under the Marine Mammal Protection Act, which is the act of harassing, hunting, capturing, or killing a marine mammal or attempting to do so. So that means that entanglement events cause problems not just for the animal itself, but for people too. Entanglements and other takes can lead to entire fisheries being shut down, as happened to the California Dungeness crab fishery in 2019 following a 2017 lawsuit. And unfortunately, the rest of the West Coast, including Oregon, is no exception to this problem. So as we all know, the state has an extremely productive and lucrative Dungeness crab fishery, which relies on fixed gear, including pots and ropes. And entanglements have occurred in Dungeness crab here, as well as active and derelict gear from other fisheries. The way that Dungeness crab gear is tagged makes it more trackable than many other types of gear, allowing the collection of data like we'll see here. So this figure was produced by the Oregon Department of Fish and Wildlife. And on the x-axis, we show time, and on the y, it shows the number of whales entangled in Oregon Dungeness crab gear per year. This blue color indicates humpbacks, and the green is for gray whales. There have been 13 confirmed entanglements since 2003, and this graph focuses on an 8-year period, which saw a sharp increase in entanglements, especially for humpbacks. Years like 2013 and 2016 with no bar had no confirmed entanglement events. And at first sight, it might not look that concerning that a whale or two are entangled every year. But however, the vast majority of whale entanglements go unreported. And so for every reported event, there are likely many others. And some research has estimated that reported entanglements represent only about 10% of the total. Also, it's important to remember that these takes are illegal under the Marine Mammal Protection Act and the Endangered Species Act, which protects threatened humpback, blue, and fin whales off of Oregon. So we know that each of these events presents legal and financial challenges for Oregon and for communities. So the stakes are high, and there are numerous efforts being undertaken to reduce the risk of entanglements to whales, including multi-pronged management efforts by the Oregon Department of Fish and Wildlife. These include supporting research, monitoring that they can combine with adaptive measures that allow them to respond within a season, voluntary accordance by fishermen with best practices, technology development for improving gear, and education and public awareness. And as part of these efforts, they're also putting together a conservation plan and incidental take permit application, which would allow the occasional take of a whale without shutting down the fishery. And that application is in the process of drafting and feedback right now. There's also been a great deal of problem solving and collaboration by the Oregon Whale Entanglement Working Group. When it first started working together in 2017, this group identified a major knowledge gap. We don't know where to expect whales to be in Oregon waters during the Dungeness crab season. And so that question is the impetus for the work that I'd like to discuss today, which uses ecosystem modeling to learn where and when to expect whales, so that we can help inform these management efforts to mitigate entanglement risk. So one of the central tools that we use to predict whale distributions are species distribution models. And this is just a quick look at how they work. So we feed inputs into a mathematical model, such as sightings of whales off the coast of Oregon, and information we think is important to the whales, like water temperature, bottom depth, and levels of their prey. And then a lot of fancy math happens in that model, and it spits out predictions of whale occurrence in time and space. In order to check the model, we use data that the model has never seen to assess its performance and make adjustments as necessary. So what does a species distribution model look like? This is a question that I had for about the first year of my PhD. So I wanted to show just a look at that today. And the answer is that it looks like a lot of code. This isn't my code, but it gives you a flavor of what this process looks like. It's basically a lot of text and symbols and letters that become these wiggly lines. And no need to, take too close a look at this, but basically this gives us a look at how the different input variables like temperature and salinity impact the whale distributions that we see. And so what is the process of modeling like? I also had a lot of questions about this. Basically, it's a lot of sitting at your computer and coding. And this project is the first time that I've learned how to code. And I was really intimidated by it at first, but I've now found it really empowering to learn how to do this. So lots of time with the squiggly lines, a lot of chocolate when your code doesn't work and you're not sure why. And lucky for me, a lot of collaboration. So I can't emphasize enough that all the work I'm describing today is a huge team effort by so many incredible people. And I wrote this blog post recently called "It Takes a Village to Raise a PhD Student" at a time when I was feeling particularly overwhelmed with gratitude for all of the people, which include formal collaborators, my advisors and committee members, and even strangers who have taken time to meet with me and help me figure things out. So now let's take a closer look at what these model outputs look like. So these figures show the Oregon coast. You can see Newport for reference, and the land in these figures is black, and it's the ocean that has color here. And you can read these like a heat map. So on the left here, so all of these show the predicted whale distributions in September 2021. And on the left here are blue whales, in the center are fin whales, and on the right are humpback whales. And reading this as a heat map, you can see that the color indicates the number of predicted whales per kilometer based on the environmental information that we gave the model. And if you see these little points on each figure, these white points that are outlined here, those are part of the process of validating our models with an independent data set, like I mentioned before. And these include opportunistic sightings and citizen science. And you can kind of see at a glance here that some points are consistent with our predictions and others are less so. And this really emphasizes points where our models perform well and also how they can be refined. And that is why we want to bring in parade data and help better align our predictions. So ultimately, layering maps of fishery effort over our model predictions can help identify areas of overlap between whales and fishing gear in time and space. And these modeling efforts are part of Project Opal, which stands for Overlap Predictions About Large Whales. The goal of this project is to provide the best predictions of where whales will be and will not be, so that managers can make spatially targeted decisions about potential fishery closures, therefore minimizing burden to fishermen while also maximizing the protection of whales. So you probably have the sense that these models rely on a lot of data, which isn't that easy to get when we're trying to study animals that live far from shore often and underwater. But lucky for us, getting that data tends to be pretty fun. So this is the head of the GEM lab, Dr. Lee Torres, conducting a helicopter survey with the US Coast Guard. These surveys are a major source of whale data for our models, as well as ship-based work, in which observers survey with binoculars and record their sightings. I also want to talk about the Northern California Current Ecosystem Cruises, which are another really important data source for us. These are run by NOAA, and they've been going for 20 years, so they represent a really rich and wonderful time series of data. And we've had marine mammal observers join since 2018. They're fabulously interdisciplinary. There are people aboard every cruise studying phytoplankton like the diatoms that we looked at before, as well as harmful algal species, which have been in the news a lot in recent years. People studying the zooplankton community. A seabird observer has started joining recently. And of course, we're studying whales with them. So I wanted to share the experience of being at sea with you a little bit as well, because being out there in this incredible environment, looking for whales all day with amazing colleagues like these is one of the biggest joys of this work for me. I was lucky to join one of the NCC cruises in May 2021. And this is us heading out. You can see we have our pandemic protocols in place, but we're really excited under our masks and very energized to be out there and look for whales. And then here's a picture from day 5 where I'm trying to sleep during a fire drill because it turns out that staring at the ocean for 14 hours a day is exhausting. But like I said, being out on the water and working with such a great team is amazing. And I truly enjoyed this entire experience. So that's how we collect data on whales. But how do we study krill, which are tiny, they never announce themselves by breaching over the water, and they're even harder to see. So this ship-based work is really crucial. This is the Bell M. Shimada, which we use for these Northern California current cruises. And we use net tows when we're aboard. So nets like this on the right let us collect samples of the zooplankton community at a given site and learn about the abundance and diversity of that community, as well as the body condition and details about the individuals that we catch. So krill have a really important behavior that I want to talk about for a minute, which is called diel vertical migration. So that means that they tend to occupy deep waters during the day to avoid predators, and then they migrate to the surface at night to feed. And this graph, which it looks like I forgot to cite, but is by the Bermuda Institute of Ocean Science, and I'm happy to send a link if anyone is interested in more about this resource. This graph shows time here along the x-axis and then depth on the y. And you can kind of see moving through these different parts of the day that as our zooplankton change their depth vertically within the water column, sampling at different times of day looks really different. So that means that for a given point in the ocean, sampling at day or night can give you a really different picture of the zooplankton community. So when we want data about krill, we use these nighttime tows that capture our krill community when they're at the surface and easier to access. So a little bit about our 2 krill species off the coast of Oregon. This is Thysanotessa spinifera on the left, which we call T-spin affectionately, and Euphausia pacifica on the right. And for whales, not all krill are created equal. There are some really important differences between these species. So T-spin tend to be a bit bigger and they're fattier, and they live closer to shore. EPAC tends to be a little bit smaller. They're still quite fatty, but less so, and they live farther offshore. And in general, EPAC is more abundant and more broadly distributed, and T-spin, and T-spin is less abundant, and their distribution tends to be patchier. And so because of these differences, these 2 species are likely to respond to climate change differently. And that will be really important to whales and all the other predators that we've talked about today. So one of the main ways that we study individuals and how they're changing is by collecting samples using nets like we just looked at. And there's also another very cool method, and that is by essentially listening for them in the ocean. So this is a picture of me from a cruise on the research vessel Pacific Storm this last October, looking at data from an echo sounder, which is basically a fancy fish finder, and using that to look for krill. So this is a really special type of acoustic work. We can send sound into the water and listen to what comes back. And this is how it works. The echo sounder emits pulses of sound at a known frequency, and then it listens for their return after that sound bounces off the seafloor or things in the water column. And based on experiments in the laboratory, we know to expect our krill species, EPAC and T-spin, to return those echoes at a characteristic decibel level. So by constantly pinging the water column with this sound, we can record a continuous acoustic profile along the cruise track of the vessel and analyze it to identify the features and the animals that we recorded. I'm going to take a sip of water, and then I'm very excited to look at some echograms with you. So I've spent a lot of the last few months looking at this type of data, these sonic records of the water column. And so I wanted to look at a few with you today. So just to get oriented, you can see this black bar on the left shows that we're at about 20 meters of depth towards the top there. And there's this whole layer of scatterers at the surface of the ocean above that depth. And from what we know about the frequencies that are returned there, we expect that this blue layer is copepods, and possibly at the top above that with the red and the green color, that represents krill. Now we're a little bit deeper in the water column. So you can see that the seafloor here is at 114 meters. And this view, which is from the same cruise in March 2020, shows these really interesting vertical aggregations in the water column. And this kind of gives you a sense of the type of data that we're able to extract here. So by basically running these echograms through a series of filters, we can get the size of swarms like these, their density, their depth in the water column, and other really important information that lets us learn about krill and the characteristics that describe their swarms. And this is also from that same March 2020 cruise. And I think this is an especially cool moment in time that was recorded. So you can see at the top, again, we have that surface prey layer. And then moving down there, these little mid-water column swarms at about 50 to 70 meters depth. And then when you move down lower, you see this really deep aggregation. And preliminary results from the work that I'm doing right now are suggesting that whales may actually preferentially target these types of deep aggregations, which we see here, which is something that my advisors and I are really curious about. So now I want to show you just one final echogram. And this is something else that I find really exciting. So last year, a colleague who does this type of work mentioned that when she was at sea looking at live outputs from her echo sounder, as they were sailing past an area with a lot of foraging blue whales, she noticed something really unusual on her echogram. And that was that the swarms of krill had these holes in them, which she thought looked like maybe the krill had arranged themselves around an object, or maybe something had come along and taken, a bite out of the aggregation. So I thought that was very cool when she told me about it. And then I was so excited to be processing data. And this is the same March 2020 cruise where we saw these curious holes in our aggregations, which I'd never seen before. And that's something that I'm really interested in learning more about. So what's next with this research? From this acoustic data that we just looked at, you kind of get a sense that we're able to learn a lot about krill swarms at both fine and broad spatial scales, and potentially even detect signs of whale foraging. So I'm really excited to continue with these analyses that I've been telling you about, and also to go to sea more and learn about interactions between swarms and these whales. So I am headed out on the next California Current cruise this May, along with two of my lab mates. And we're going to do something different and really fun here, which is that as they collect marine mammal surveys, I will be looking at the echo sounder data and conducting real-time prey observations, so we can learn more about intersections between krill and whales as the relationships unfold right in front of us. And as this data set grows over the next several years, I'm really looking forward to working more with this concurrent predator and prey data and feeding that into our species distribution models in order to make them even more powerful. Something else that I'm really excited about is in this upcoming August, I'll be headed along with the rest of the GEM Lab to the Society of Marine Mammalogy Conference in Florida to present about this work. And I'm able to do that with funding from the ACS Oregon chapter. So thank you so much for your support. I've been talking all through this presentation about my amazing lab mates and the work that they do. And so if you want to learn more, I wanted to share a few opportunities about that. This is a website that was launched recently called Individual Whale, which gives you an up close and personal look at some of the really interesting gray whales that frequent the coast of Oregon here. And I also mentioned the blog where we share about our current research. And that's a really fun way to kind of stay up on what people are working on and thinking about. And in addition, the Zooplankton Ecology Lab has a great website full of blog posts and amazing pictures, as well as outreach and teaching resources. So here are these links, and I can put them in the chat as well. I'd like to thank my co-authors, funders, and all of the contributors who have made this work possible. I'd like to thank you for tuning in today, and I'm looking forward to taking any questions. Thank you.
Thank you so much, Rachel. That was very interesting. So let me take a look here. We have a question in the chat. Let's see here. So Paul had a question about what are some of the recommendations to reduce whale entanglement?
Yeah, that's a great question. And I think that is a really kind of multi-pronged effort by a lot of different groups. So just off the top of my head, some of the kind of diverse measures that have been put in place include slowing ship speeds in traffic lanes. Like one famous, case about that is trying to prevent ship strikes with blue whales in Southern California and mitigating speeds as part of that. And then, it's my own personal bias, but I think this work that really emphasizes spatial and temporal co-occurrence is such an important part of trying to reduce entanglement, because at the heart of that, right, is the desire to protect both whales and coastal ecosystems and also human communities, and try to figure out how those can safely coexist. So I see a lot of interesting potential there.
And as part of the whale entanglement working group, we did come up with some recommendations to help reduce whale entanglement. Oregon Department of Fish and Wildlife submitted their conservation plan for public comment, and we did submit our public comments, and hopefully some of you did as well. And currently they are working on revising that draft conservation plan before they submit it to National Marine Fisheries Service. We're hoping that we will get a chance to take a look at that plan prior to their submission, but a lot of the things that they were looking at were ways of trying to reduce risk of co-occurrence, which means fishing in the same areas the whales are. They have already taken some steps. The first round of measures that were put in place were more along the lines of gathering data. So it was marking, it was some information from the fishermen about where their fishery is. So kind of more of a data gathering process. Then the second set of measures is decreasing number of pots by eliminating replacement tags. It involves changing the depth restriction for crabbing later in the season to help avoid being in humpback habitat. So there's a variety of measures, and then they've included some other new measures in this plan. So we're looking forward to seeing that. Let's see, we have another question. Can you please elaborate on the process how the Earth's rotation causes upwelling?
Yeah,
Maybe show
totally.
that slide again.
Yes, let's see. Yeah, exactly. Okay, so so kind of looking at this upwelling diagram again. So these dominantly northerly winds that start in the spring, those essentially drag the surface of the water with them. And the Coriolis effect, if that's familiar to people, describes the deflection of objects because the Earth is round and because there's an inherent rotation instead of something traveling in a straight line. So it's the Coriolis effect is why this water ends up becoming displaced offshore as this curvature kind of builds up while the wind moves from the north. And that's called Ekman transport, is this offshore movement of this layer of water. And that creates a pressure gradient because you have, you basically induce this slant into the surface of the water where you have more water offshore and less inshore. And we know that water doesn't like to sustain a gradient like that. And that's what pulls this water up from depth.
Okay, we had another question. When we had the dead zone here around Hecata Banks, how did that affect whale feeding distribution?
That is a great question. And I don't know specifically about that, but I would be really happy to talk with people who know more than I do and send information about that if you'd like.
Okay, that would be great. And I think Clara, when she talks to us next month, she probably will have more information on that since she's studying the whales in the local area.
Yes, absolutely.
So then I have a question. Is there another source for reporting sightings of whales directly to you? I use the Whale Alert app, and I know Dr. Torres sees that. I have seen blue, say, fin, humpback, minke, as well as gray whales from our shore sites. So is there any other way besides the Whale Alert app to get information to you?
So as far as I know, Whale Alert is the best direct source. And I actually just saw that my advisor, Lee Torres, has chimed in about the chat with this, which is great. And also a note that the Individual website that I showed at the end will have a feature in the future that allows people to input sightings and photos as well, which will be really cool. So thank you so much for wanting to contribute to that validation data set. It's really valuable and we appreciate people sending those sightings in.
Wonderful. What do you mean regarding knowing gray whale migration during crabbing season in Oregon? We know a lot of it. Are you getting info for legal submission? Well, with this project, it's looking more at the endangered species. So we're looking at bluefin and humpback whales, not necessarily gray whales. Is that correct, Rachel?
Yeah, thank you for pointing that out, Joy. That's a really important point, that for the models that I described, those are exactly for those protected blue humpback and fin whales. I'm not modeling gray whale distributions there. And you're right that there's a lot of great data out there about whale distributions. And I think that the need that was pointed out, that's really the impetus for this work, is this like much more kind of constraining our distribution expectations more in time and space so that we can most effectively expect where overlap may occur and give that information to managers.
Okay. And Lee has put some information in the chat. Yes. The crab pots being less than 40 fathoms, starting on May 1st of each fishing season. Let's see. What is the typical speed of the southbound California current, and at what distance offshore?
Wow, that is such a great question that 6 months ago, me in my coastal oceanography class would have done a much better job with. If I remember right, we're in the ballpark of 10 meters per second, but that's another thing that I would like to look up and get back to you on to make sure I'm not saying anything misleading.
Okay. Are you looking at using what's known as ropeless or acoustic gear for your crab fishery?
Yeah, I'm so glad that you mentioned that, because that's something that I didn't touch on and I wished I had in the question about how to avoid entanglement. So that's not something that I am working with personally, or that we are on this project, but I think that's a really important direction that has people, in the Atlantic who are really working on this issue to try to mitigate entanglement risk with North Atlantic right whales, and then also collaborating with people who are working here in the Pacific to try to learn. We know that lines and weighted gear can be tricky for whales that are navigating around it. So are there ways to change that technology to help make that less risky? And my understanding is that right now there's a lot of development, and things are still pretty expensive and not quite ready to be rolled out on a broad scale, but that's a really interesting direction for research and development.
And I will just chime in a little bit on that. So our environmental NGO coalition has been recommending that they begin to look more seriously at using some of the on-demand gear, the acoustic gear for the fishery. And so I think that's more on the environmental conservation side than on the science, as far as promoting that to Oregon Department of Fish and Wildlife. Okay, and then Lee had mentioned, yes, using the Whale Alert app was very important for contributing data, and that they'll also be adding to the website Individual to report gray whale sightings and photos in the future. We have somebody saying, thank you so much, Rachel, for this fascinating research. Go whales. Um, and thank you so much for your time this morning. Very informative. Do we have some other questions or comments for Rachel? I would like to say thank you. It was an excellent presentation and very interesting, and will be interesting to see where this krill kind of fits into the maps.
Yeah, thank you. Thank you so much. And I agree. I'm really excited to see that side of things develop as well.
I think the more different pieces we pull from to put into these models, the much better understanding we will get by using all these different methods and pulling them together. Okay. Then we have some comments here. Thank you for all your work. Thank you for sharing your interesting research. Great presentation. What a great talk title, too. Excellent presentation. Thank you for sharing your work. Great talk. Thanks. So, all right.
Well, thank you so much, everyone. And thanks for tuning in and sharing your Saturday morning with me.
And enjoy the Society for Marine Mammalogy conference.
Thank you.
All right. Anybody else have any questions? You can feel free to unmute and unmute yourself if you have some further questions.
It was interesting. Thank you.
Oh, thank you so much. I appreciate it.
So, did you apply again for another grant for any studies?
No, not since last year. Originally, this Society for Marine Mammalogy conference was going to be held in 2021. So it's really nice that I'm able to, still have this funding to attend now in 2022.
Mm-hmm. Very good. Yes. And when you go on the NOAA cruises, the Northern California cruises, is that funded through Marine Hatfield Center or through OSU? How do you pay for that?
That's a great question. I think that the funding to send me comes from the broader funding for Project Opal. And I think there are different sources for all the different types of researchers aboard.
Mm-hmm. Good. Yeah. And so are you based at Hatfield?
Yeah, and like I mentioned, something that's really cool about my PhD experience is that I'm in two labs, the one based at Hatfield here, the GEM lab, and then the zooplankton ecology lab is in Corvallis at the main campus. So I kind of get to bounce back and forth between the two locations.
Mm-hmm. Good. So you mentioned on the zooplankton and the two species. So the gray whales normally feed in closer. And so what does it make a difference on distance from the shore and which species is prevalent? Is it depth? You said that the the T-spin is not as abundant, but
yeah, exactly. So they have really there's definitely a lot of variability in their distributions along the shelf and across it. T-spin tends to be over the slope more, and EPAC a bit further off. And so something that's also really special about research in this area, you might have heard of the Newport Hydroline cruises that happen. And those have been going for, I think, something like 30 to 40 years, which is a really fabulous long time series. And by studying the zooplankton community over that long time, those data have shown that about 15% of the total krill population off the coast tends to be T-spin, and 85% is EPAC. And so that's really interesting to us in a lot of ways, because it kind of brings like the quality versus quantity question in. So we know that EPAC is great prey for whales, and that's 85% of the krill that's out there for them here. But they really seem to target sometimes that 15% of fattier, even more nutritious T-spin. Yeah, thanks for the question.
So if I may embellish on that, if we go like up to Alaska, where we have the colder waters and the upwellings where the krill is up higher, is the percentage similar?
Ooh, you know, so I don't know as much about the zooplankton community up there, which is funny since that's where I'm from. But I would say no, it's not the same. I think the zooplankton community looks pretty different. And that's actually a really exciting future research direction of the GEM Lab, is to be looking more into whales in the Gulf of Alaska. And so that might be something we're learning about more over time there.
Thank you.
Thanks.
Okay, well, thank you very much, Rachel. We are so glad to have had you speak with us today. And so with that, unless anyone else has any other question, we'll go ahead and end the meeting.
Thanks so much, everyone.
Bye.